All right, let's jump right in. Imagine someone dumps billions of letters of raw DNA code right onto your desk. Sounds cool, right? But honestly, on its own, it's kind of meaningless. It's literally like dumping a massive pile of bricks, lumber, and glass onto an empty dirt lot. You can't live in a pile of bricks. So today in this explainer, we're going to transform that raw data into a highly structured, meaningful biological blueprint. We are talking about the absolutely fascinating science of genome annotation.
Here's our quick roadmap for today. We're going to cover decoding the blueprint, the 2 types of annotation, the 6-step process, tools of the trade, real-world applications, and finally, we'll look at the future of genomics.
So let's kick things off with Section 1, Decoding the Biological Blueprint.
Successfully sequencing a genome is a massive achievement, for sure, but actually, it's really just gathering the raw materials. So what happens after the sequencing is done? How do we take that chaotic pile of genomic materials, that endless, dizzying string of A's, C's, T's, and G's, and actually build a structured, understandable house out of it? Well, you can't just leave it in a pile. You need an architect to come in, map out the floor plan, and label every single feature to give it an actual biological purpose.
In that architectural process, that is what we call genome annotation. Basically, it's the computational and experimental process of identifying the exact locations of genes and other genomic features and then assigning functional information to them. This is the fundamental, absolutely non-negotiable step that transforms raw sequence data into actual, usable biology. Without it, you basically just have a really long, incredibly boring book written in a language that literally no one can read.
Moving right along to Section 2, the 2 types of annotation.
Now, drawing our ultimate biological blueprint actually requires 2 completely distinct layers of information. The first is structural annotation. Think of this as finding the walls, the doors, and the hallways of our house. It identifies the physical features of the genome. We're talking protein-coding genes, exons, introns, start and stop codons, promoters, the physical stuff. But then, we need functional annotation. This is the part that determines whether a room is a kitchen or a bedroom. It assigns the actual biological meaning. It predicts things like protein function, metabolic pathways, and cellular locations. You absolutely have to have both of these layers to truly understand the organism.
All right, Section 3, the 6-step process.
So, how do we actually do this? Well, here is the step-by-step journey. Step 1 is genome sequencing, just getting that complete raw DNA string. Step 2 is quality assessment, where we meticulously clean up the data and toss out any sequencing errors or contaminants. Then comes Step 3, gene prediction, which is where we hunt down potential genes. Step 4 is structural annotation, mapping out those coding regions and regulatory elements. Step 5 is functional annotation, where we cross-reference our newly found genes with existing biological databases. And finally, Step 6 is validation. This is where we actually step away from the computer and use real-world experimental lab methods like RNA sequencing or proteomics to confirm our computational predictions. It's a highly rigorous pipeline for sure.
Let's actually zoom in on Step 3 for just a second. Finding microscopic genes hidden in billions of base pairs, well, it isn't done manually by some tired researcher with a magnifying glass. No way. We use powerful computational algorithms. You can kind of think of them as robotic builders on our genomic construction site. They hunt down and locate potential genes using 3 primary methods: ab initio, homology-based, or RNA-seq-assisted prediction.
Okay, so ab initio prediction basically attempts to read the code entirely from scratch. It relies purely on the mathematical and statistical characteristics of the DNA sequence itself. And it's fantastic because it doesn't need a reference genome at all. But the downside? It definitely has lower accuracy for highly complex genomes. Alternatively, homology-based prediction cleverly relies on, well, cheat sheets. It compares your unknown sequence to previously annotated genes from related organisms. And because nature loves to reuse a good design, this method yields much higher accuracy and super reliable functional predictions. Oh, and RNA-seq-assisted annotation? That uses actual gene expression data to find actively transcribed genes, which is also highly accurate.
Which brings us to Section 4, Tools of the Trade.
You wouldn't use a hammer to drive a screw, right? Well, bioinformatics researchers don't use the same software tool for every genome. BLAST, for example, is our absolute go-to for sequence similarity searches. PROCA, that's a rapid software perfectly tailored for bacterial genomes. But what if you need to predict genes in a complex eukaryotic genome, like a plant or, a human? For that, you'd use Augustus. Maker is this incredible integrated pipeline that combines multiple sources of evidence together, and RAST is a fully automated server specifically designed for microbial genomes. Basically, it's all about picking the right tool for the specific organism you're looking at.
But here's the thing. Finding the structural gene is really only half the battle. To assign true functional meaning, researchers have to cross-reference their findings with massive global biological databases. We're talking heavy hitters here, like gene ontology, the Kyoto Encyclopedia of Genes and Genomes, often called KEGG, UniProt, FAM, and Interpro. So when our software pipeline finds a new gene, it essentially queries these giant global libraries and asks, hey, have you ever seen this sequence before? And if so, what exactly does it do?
Next up, Section 5, Real-World Applications. So why does mastering this blueprint actually matter outside of a computer lab? Well, because it directly impacts our daily lives and, frankly, our survival as a species. In medicine, it allows us to identify disease genes, which enables personalized medicine and helps diagnose complex genetic disorders. Over in agriculture, it's literally the foundation for engineering disease-resistant crops to help feed our growing planet. It drives biotechnology through metabolic engineering. In microbiology, it helps us rapidly identify dangerous pathogens and track antibiotic resistance. And it even allows evolutionary biology to trace how species evolved over millions and millions of years. I mean, it is the absolute bedrock of modern biology.
And finally, Section 6, the future of genomics.
Now, despite having all these amazing tools, the computational challenge we're up against remains completely staggering. We face massive genome complexity and highly repetitive DNA sequences. Just imagine trying to read an entire library of books, where all the words are mashed together without any spaces, and half the sentences just repeat the word "the" 1,000 times in a row. Sound like a nightmare? Yeah, it leads to prediction errors. Add in the fact that real-world lab validation is slow and expensive, and that these databases require constant, non-stop updating, and you can quickly see why this is a monumental, almost never-ending task.
But here is where it gets incredibly exciting. Just look at the recent advances driving the future of this field. Artificial intelligence and machine learning are dramatically, and I mean dramatically, increasing the speed and accuracy of our predictions. We now have long-read and single-cell sequencing technologies pulling richer, way more accurate data than we've ever seen before. Cloud-based bioinformatics platforms are allowing researchers all over the globe to crunch massive data sets, integrating multiomics, which is combining genomics, transcriptomics, proteomics, and metabolomics all together. We are rapidly moving toward a world of real-time annotation for newly sequenced genomes.
Which leaves us with a fascinating, highly personal thought to chew on. We are rapidly approaching an era of true precision medicine, driven by the exact technologies we just talked about today. Fully automated, AI-assisted genome blueprints are legitimately on the horizon. So when AI eventually creates your fully automated, personalized genome blueprints, what will it reveal about your health, your ancestry, and your future? It's definitely something to think about. Thanks so much for joining me for this explainer. I really hope you found it as fascinating as I do, and as always, keep learning.